Dear students, before you start reading the eyeball, its layers and the intraocular structures, for a better understanding, if you follow the dissection of cow's eyeball, it will help you in understanding the eyeball structure. Now, what you are seeing here is the cow's eyeball, and this is the anterior most part. You are seeing the cornea, and this is the corneal margin, and this part is the sclera. The outermost layer of the eyeball is the sclera posteriorly and cornea anteriorly, and surrounding it you are seeing the extraocular connective tissue, the fat and the muscles that get inserted into the sclera. Then if you see the posterior part of it, what you are seeing is the optic nerve, so which is coming out from the posterior pole of the eyeball, and these brown colored ones are the extraocular muscles, and this is the extraocular fat. Here you can see the extraocular muscles and optic nerve more clearly after trimming the structures outside the eyeball. Then, so the eyeball is kept in its position by the extraocular muscles and the extraocular fat, adipose tissue. And then to maintain its shape, it contains the fluid inside, that is the aqueous humor and vitreous humor. Now, if we make a cut around the sclera behind the cornea, all around the eyeball, the aqueous humor will come out. Now, the aqueous has come out and it is here. You are seeing the thin fluid. Now, it is made into an anterior segment and a posterior segment of the eyeball. And this jelly-like substance which you are seeing is the vitreous humor. Here also you are seeing it in relation with anterior segment. And this layer which you are seeing is the retina, and this dark one is the choroid, and this thick one is the sclera. Here also you can see the vitreous, then you are seeing the lens. Now we will see the posterior part of it by removing the vitreous and then drying this area. You can observe the retina, and there is a depression here. This is called the optic disc. So the retina is the nervous layer. It consists of cells and the axons of which will be passing through this optic disc and will come out from the posterior pole of the eyeball as the optic nerve. And here you can move the retina. So it can be pushed towards this optic disc where it is attached. So the rest of the part can be moved up to this. You can see it all pulled towards the optic disc. Now you are seeing a colored area. This is called tapetum, which is seen in animals, which facilitates night vision for the animals. Then this is the dark one is the choroid. This is the choroid, and what you have pushed is the retina. So now you can see the tapetum, the retina, and then the choroid. The outer one is the sclera. This is the optic nerve, posterior pole. Then, so the one that is pushed with the probe is the choroid, the dark one, which is a vascular layer. And now you can see the fibrous of sclera, which is also keeping the shape of the eyeball. This is the sclera and this is the choroid and this is the retina. Now you can see the anterior part of it where you are seeing the lens and then you are seeing the dark one around. This is the ciliary body with radiating processes you are seeing. And if you remove the lens and keep it aside, then see the one. This is the one where you have kept the lens back. And you can see the choroid here, the ciliary body you are seeing. See the lens, the choroid and ciliary body. This is the retina you are seeing in relation with the anterior segment. Then once you remove the lens, you are seeing another smooth dark one. This is called the iris and this is the pupillary margin you are seeing. So you can identify the retina, the choroid, the ciliary body and the iris. Now you can see the pupillary margin clearly and the iris also. This is the iris and this is the ciliary body. Now this is the iris and then the probe is touching the posterior surface of the cornea. So this is the anterior surface of the cornea. Now let us see what is anterior chamber and what is posterior chamber. So if you can identify the iris, between the iris and cornea is the anterior chamber. The probe is in the anterior chamber and between the iris and the lens is the posterior chamber. And if you keep the lens in place, now identify the posterior chamber is between the lens and the iris, and behind this lens, posterior to the lens, what you will be seeing is the vitreous humor or vitreous body. Now you could understand the layers of the eyeball and what is optic disc and the various chambers, the aqueous humor and vitreous humor. Now you can understand the structure of eyeball.